we're officially over time, but we have one more speaker. Hi, Patrick Gelbach is joining us.
Okay, thank you all so much for being here. I'm super grateful for the chance to talk to you about my work, which is in metabolic modeling of macrophage subtypes in colon cancer. And so in general, in cancer, we know part of the difficulty in treating the disease is not just the cancer cells themselves, but the fact they exist within this complex microenvironment consisting of different cell types, such as stromal cells, immune cells, different extracellular metabolites that are secreted by those cells that all come together to affect the way that these cancer cells behave and can really collectively be viewed as active promoter of cell disease. And so the most common type of immune cells in the tumor microenvironment, and ones that I found particularly fascinating, are these macrophages, which can take these broad, two very, very different phenotypes. So broadly referred to as M1-like and M2-like. The M1 are pro-inflammatory and the M2 are immunosuppressive. So in general, you would see a proliferation of M1 cells in like a bacterial infection where the immune system has to ramp up and respond and kill those foreign invaders. You would see a proliferation of M2 cells in following wound healing and other tissue repair, where you really want to avoid being like an overactive immune system. And so it's fascinating that the same cell population can take on wildly different behaviors depending on the context. And cancer exploits this M2-like state because the immunosuppressive aspects and the angiogenesis promoting aspects also promote cell survival for the cancer cells. One, I think, increasingly appreciated aspect of this M1 M2 paradigm is that the cells exhibit differences in preferred nutrient sources, differences in the ways that they make energy, and then differences in the ways that they use that energy. And so metabolism, I think, can be seen as a driver of phenotypic differences, or really, like, more softly could be said as the way that these cells make and use energy affects what these cells do. And we care a lot about differences in how these cells behave. And so, like everyone here, we've decided to use, the focus of my work is to understand the metabolic differences between M1 and M2 macrophage subtypes. And so to give a little roadmap for what the way this will progress, we want to first use patient data to generate out these M1 and M2 specific models. We then want to identify the main differences both in network structure and in predicted activity. And then we want to really apply the models to identify new treatment strategies. And so, like everyone here, we're using modeling of this system, and most of metabolic modeling really boils down to biology's central dogma, where genes are transcribed to RNA, translated to proteins. Those proteins carry out the interconversion of one metabolite to another through a series of metabolic reactions. And when you just look at that relationship, you can really define what you're most interested in and construct a metabolic network, which is a series of connected biochemical reactions that collectively convert metabolites between themselves. And so you might recognize this figure of central carbon metabolism, where of glycolysis, TCA cycle, and pentose phosphate pathway. But in reality, there are hundreds of metabolic pathways and tens of thousands of reactions that happen. So the traditional way that I think most people here have done today is with very detailed ODEs, very detailed kinetic equations, where you focus in on one specific, in our case, conversion of one metabolite to another, represent it with a differential equation, and that differential equation relies heavily on distinct parameters to contextualize the model. And that is nice because it allows you to predict the change in metabolite over time. However, one big problem is that as you try to model more and more reactions, you end up getting more and more model parameters, and therefore you either need more and more data, or your metabolic parameters are no longer identifiable. And so you there's this discrepancy between the size of known metabolic networks and our ability to use very detailed kinetic equations to approximate their behavior. And so on the bottom right is a small network, or is a relatively large ODE network that we published of about 65 metabolic reactions. As I mentioned, a cell can have up to like 10,000 or more metabolic reactions. And so there's this discrepancy between very detailed kinetic parameters and the real true amount of reactions that go on in a cell. And so an alternative is what's commonly referred to as top-down modeling, where you still use the known gene protein reaction metabolite relationships, but instead you simply define them as an input-output relationship. And so you take an input metabolite converted by an enzyme catalyzed reaction to an output metabolite, and an additional little bit of information is what genes are responsible for carrying out that metabolic reaction. And so for our previous example, glucose is converted to G6P through the glucokinase reaction, which is linked to the hexokinase 4 gene. And you can use these for all known metabolic reactions, and therefore represent metabolism as a series of linear equations. And so these models, when you do it large enough, are called genome-scale metabolic models, and consist essentially of two different matrices. One is the stoichiometric matrix, which has the reactions on the y-axis and metabolites on the x-axis, and each individual coefficient refers to whether or not that metabolite is used up or produced by that particular reaction. And then because we have the linked genes and proteins, we also have this Boolean network describing gene-protein reaction relationship rules. And the thing that I like about this is that in particular allows you to integrate different types of omics data into a single contextualized framework. So these models can act as scaffolds onto which we can overlay data. And so that's generally what people do, is they take a generic stoichiometric matrix and then use those gene protein reaction rules and context specific data to generate out a context specific stoichiometric matrix. And so we use the Recon 3D model because we have genes, proteins, and reaction and metabolites linked. You could use transcriptomics, metabolomics, fluxomics, really multiple different data types to generate out this condition-specific model. We're mostly focusing on transcriptomics data, and there are a range of tools that do that integration of transcriptomics onto a generic model, depending on what facet or what aspect of the input data you really appreciate. So like you want to maximize the presence of highly expressed reactions is one technique. You could also try to pare down intensely the lowly expressed reactions. And so there are different tools that do this overlay of data onto a generic model to generate out a condition-specific model. The nice thing then is that you can use that stoichiometric matrix to predict the flow of the flow of material through the network, essentially getting at the behavior of the cells. And not to go too deep into the math, but the stoichiometric matrix times the distribution of fluxes equals the change in metabolite concentrations. If you assume you're looking at a single snapshot in time, or assume that the system is at relatively steady state, that right-hand side goes to 0 and lets you solve for the distribution of fluxes. And so you get this is a toy network with the darkened lines corresponding to higher flux through the network. And so this process of data-driven model generation and then mathematical analysis to predict a functional activity is really what we've done here. And so you can do this for really whatever question you're interested in. You could look at mutation state of different colon cancer or different cancers. You can look at male, female patient, different time points. In our case, we're looking at M1, M2, and I mentioned there are different tools to do this. Data overlay, we didn't know which one to choose and didn't really want to bias the results, so we just tried them all. And each, even with the same base model, the same data, and different tools, you get each approach generates out a single model. And so we got an ensemble of metabolic models that all have integrated that data to generate out a M1 or M2 specific cell. And we consolidated those high confidence metabolic components into M1 and M2 models that we had high confidence in their construction. And so we can then compare network structure so this is like a Venn diagram showing the number of model components that are unique or shared. And so as you would expect, a majority of the metabolites, reactions, and genes are shared between these M1 and M2 models. Those probably correspond to just generic cell function or generic macrophage function, but we think that the ones that are distinct, the red and the blue, probably correspond to M1 or M2 specific activity. And so we can look at the metabolic pathways that are represented by those differentially present or absent model components, and we see amino acid metabolism and inflammatory pathways come up as present in the M1, and fatty acid metabolism come up as M2, which is kind of a sanity check that we're getting reasonably supported information as a result of this data generation, model generation from the data. And then can then predict flow of material through the network. And we used a flux sampling approach. Another technique is called flux balance analysis. But in our work, we wanted to see what how material flows through these networks as different between the M1 and M2 state. And we paired our predictions of flux with a little bit of graph theory. So this is showing a flux weighted network centrality, which in our perspective, we're taking to really mean importance in the M1 state or important in the M2 state. And so the ones that are colored red are important in M1, but not in M2. And the ones that are colored in blue are important in M2 and not in M1. And so this, we're able to predict functional activity, differential activity between the M1 and M2 states. And so we see alanine import, glutamine metabolism, and TCA cyclomalic enzyme. And then for M1, and then coenzyme A fatty acid metabolism in M2. And from there, to go back to the first introduction part, there's a proliferation of M2 cells in colon cancer. And we want to know if maybe we can use our models to induce a shift towards the M1 state to get these macrophages to participate in the immune system's tumoricidal ability. And so we can take our model and in silico knock down a metabolic reaction. In our case, we decided to do pairwise metabolic knockdowns because there's some evidence that hitting two different spots in the network is more productive than just one. And this is showing the top 10 most impactful metabolic reaction knockouts. And we found this phosphate transporter and methyltransferase to induce the biggest shift towards an M1-like state. And so we can also then take our M1 model and simulate it a bunch of times and then reduce those flux distributions down to low dimensional space. We can take the M2 model and do the same thing. And then we do see that our identified intervention does seem to shift metabolism globally towards an M1-like condition. And so it's a cool way to use readily available omics data to, in the end, identify potential treatment techniques. And so in conclusion, we use these M1 and M2 specific macrophage data from patients to generate out these condition-specific models. Then we identify differences in their structure and behavior. And then we can use this to identify, to generate hypotheses to encourage a shift towards an anti-cancer phenotype. And so with that, I'd love to thank my lab and happy to take any questions. So.
So that was cool. I guess one of the things that I'm a little bit conflicted about is that the kind of macrophage state seems to be less binary and more continuous.
Yeah.
So you know, you've looked at these as being binary in some sense, and then tried to look at that through the lens of metabolism. How do you wrangle the fact that it isn't binary?
Sure. Yeah. So it's a massive oversimplification, and we really wanted to see if this pipeline works. And so I think there's probably more evidence for the M1, M2 paradigm in cancer than there is in other contexts, but you're exactly right that it's a gradient and not these set groups. And so since then, you can use the same pipeline for single cell data. And so we've been getting at that exact question of just doing this at a population level and finding out cell to cell heterogeneity and where cells fall on that M1, M2 gradient, rather than this was just first relatively broad and like approximate groups. And so, yeah, you're exactly right.
Well, a follow-up question. We published a paper a few years ago showing how acidification by glycolytic phenotype cells might push you towards more of an M1 or M2 state. Do you see anything like that in here? I mean, do you see any glucose or acidification in your pathways that would shift those states?
Yes. So I think generally what I think of is that the metabolic reprogramming is done by the cancer cells. And so like the secretion of lactate changes the microenvironment, which changes the metabolism of these M1 and M2 cells. But we assumed that they were both in the same extracellular conditions. And
yeah,
so,
but
we
do
do
see receptors for lactate, or I'm not sure what those nodes represent.
Sure.
Right? So
yeah. So our model
worth looking at.
Yes, we do. We have, I think, like a couple thousand transport reactions. And so, yes, we account for that. And we would like to eventually look at the differences in extracellular condition effect on internal metabolism.